All right. Are we on? Can you see and hear me right now? Let me know. All right, beautiful. Hello, everybody. Welcome back to Paleontologizing. I apologize for being so late today. It wasn't my fault. Starlink had a major outage, region-wide, maybe worldwide, I don't know. But just went back on, and now here we are. Welcome back to Paleontologizing. Welcome back to Eastern Utah. Welcome back to the earliest Cretaceous Cedar Mountain Formation and to a live dinosaur dig conducted with the Utah Geological Survey here. We're going to be doing some more dinosaur excavation here, but also, and I wish we had more time for this, we've gone down from 3 hours to 1 hour, but we're going to be talking with Dr. Karen Poole about iguanodonts and about phylogeny. She's an iguanodont researcher, iguanodont specialist, and phylogeny is her thing. She's currently working on a new publication trying to figure out the interrelationships between a whole bunch of different ornithischian dinosaurs. In fact, all of Ornithischia. It's going to be a big deal, and we'll see if she drops any, I don't know, any little hints about some of the groundbreaking aspects of it. I won't be surprised if she doesn't, though, because we got to keep these things, I don't know, hush-hush sometimes. Anyway, if you're new here, welcome to Paleontologizing. My name is Danny Anduza. I'm a dinosaur paleontologist. Paleontologists study fossils. I work on dinosaur fossils in particular. Most paleontologists don't really have anything to do with dinosaurs. Everything from fossil bacteria to fossil baleen whales, from fossil ferns to fossil frogs. I work on, along with bits of fossil plant. We found a nice croc tooth earlier. Yeah, all kinds of good stuff. But anyway, I'm glad you're here. Let me know if you've got any questions. The whole point of these broadcasts is to do science outreach, to like how we do our work as paleontologists, to kind of demystify all of this, you know? The paleontology in popular media and movies and TV shows and video games and stuff like that, but I think it's cool to show people how it really works. And so that's what we're doing here. Don't be shy with those questions if you've got them. Welcome to Paleontologizing. And how's Don? He is here. Let's go check in on Uncle Don over here. So those of you who were here yesterday, you saw that we had a big windstorm blow in, and then our shade tarps, some of them got lifted up. That one on the end actually got lifted completely up. One of the legs bent about 160 degrees, and it came smashing down on top of Don's finger. So he had to go to the hospital and get some stitches, and he's doing a lot better now, though. Let me show you. Let's see.
I know.
Uncle Don, they want to know how you're doing. Everybody's asking, how's Don? How's Don?
I'm doing pretty well.
Yeah.
I'm a little crabby, but
understandable. Yeah. How many stitches?
10.
10.
So yeah, pretty much went from like there to there.
Oh boy.
Shoot. I do have pictures, but probably not safe for.
Yeah. I get a little squeamish
for a family
I don't want to subject the audience to that.
But yeah, so luckily it was a quick trip to the ER, and it was a slow day at the ER, luckily, so.
Nice, they got you in right away.
Got me in right away.
Yeah. And they got to meet a paleontologist.
Yeah.
Probably made their day.
Oh yeah, that one gal was like, oh, I knew Sue Sternberg. Oh yeah. It's a small world in Moab. But.
Yeah.
And I said, well, sorry, I'm coming in looking like a dirtbag because I had like torn up shorts and covered in dirt and stuff. They go, oh, you're fine, dude.
It's Moab.
Like, yeah, it's Moab. You'd have to be a lot more dirtbaggy. I promise them. I brushed my teeth before I came in, so.
Yeah.
I try to be a respectful patient. So.
Yeah. Sounds like you succeeded. Did you really brush your teeth?
Yeah, I did brush my teeth.
So you're like, fingers bleeding? You're like,
yeah. Well, luckily, I mean, they wrapped it up right away, and then, so yeah, it was still fine. I was a little queasy for the first 10 minutes, but
yeah,
between that and the smoke, I was like, oh.
For sure. It was a weird day yesterday.
Yeah, Ethan and Fisher took good care of me.
Nice.
Dropped me off, and then, yeah, the folks were really nice there.
Excellent.
Yep, other than sticking the Novocaine needle in, everything else was pretty smooth.
Yeah, nice.
But yeah.
Well, cool.
Yeah, I was definitely going to have a battle scar from that one. So.
Oh, yeah.
So trying to take it easy today. Probably doing more than I should be. But we're heading out in the morning, so I'll have the weekend at home to recuperate.
Recover, yeah. Good timing on that, honestly. Yeah. That also means, unfortunately, that we're going to be losing Karen tomorrow. So we're going to be talking with her about iguanodonts and about phylogeny here in just a couple minutes. But before we do that, I'm sure people will be excited to see Nick Oliver is back with us. Nick from San Diego.
That's right.
He was with us in Wyoming too, and Nick's a good guy to have in camp. So glad to have him. Another news: Jurassic James left this morning, and Ken should be here in a few days. Don's gonna go home for the weekend. I don't know. People trickling in, trickling out. But yeah, we've got lots of well wishes for Don in the chat, and people are excited to see Nick too. Good stuff. Cool. So here in the quarry, I took a couple bones out this morning, and got those mapped and logged and everything. There was that one that I had gypsoned yesterday, a little medical bandage, took that out. Took this guy out over here, that weird bone chunk. And I'm excited tomorrow morning to come in here and just cut through this at a reasonable pace, not too quickly. Got to be careful to look for any chunks of bone, but I'm excited to get down to this layer so I can start removing this. I'm hoping that's where the good stuff is. That's where Fisher and Ethan are finding some stuff. Fisher's got a sizable bone right there. Rib. Pretty cool. Let's see if it works. Wow. Not too shabby. And unfortunately, it's Karen's last day with us, and we had a whole thing planned. It was going to be really cool, talking about phylogeny, and we had a whole exercise planned with this pasta, which was Karen's idea. It was really neat. We might still do some of that.
Yeah, if we've got time, we can do that.
Yeah, would you mind? Yeah, that would be really cool if you don't.
Sure.
You don't mind?
Yeah, I'm just gonna, I'm not gonna finish any of this, so
sure.
Just
be a good stopping point.
Cleaning things.
Excellent. Yeah.
But yeah, I had a good last day in the quarry. I finally found stuff.
Nice, awesome. Yeah.
Got a theropod tooth and a croc tooth, and I've got now a cervical vert coming out.
Oh, beautiful. You had a croc tooth over here too?
Yeah, like just the tip of one. It's all wrapped up now. Yeah.
Okay, cool.
Yeah.
Cool. Shoot, let me go grab the Dinosaur Frontier book real quick while you get set up.
Sure.
And I've got big gratitude to Karen for taking time out of her last day in the quarry to talk with us about phylogeny. This is a really important subject, and this is what Karen does. She's an expert on this, talking about evolutionary relationships and how we figure them out. There's a trick to it, as you'll see. This might seem a little bit arcane, but I think Karen's a pretty good teacher. We'll learn about this together. I'm gonna learn some stuff too, so I'm excited about this. Grab this book where we've got some phylogenies in it. And there you go, Arlang. Good to see you, Dad. I hope you're doing well. And these would be crocodilomorphs. Exactly, Murph. Yeah, I don't think we have any crown group crocs here in the earliest Cretaceous. So these would be crocodilomorphs and not like today's crocs. But here we go. Let's see how to do this. Take these off. There we go. Yeah, so Karen, you want to introduce yourself real quick for anybody who might be new and might not be familiar with you yet?
Sure. Hi, everybody. My name's Karen Poole. I study largely iguanodontians and other ornithischian dinosaurs. And I guess I would describe myself on that front as a phylogeneticist. So I study phylogenetic systematics, which is basically figuring out how different species are related to each other evolutionarily. In other words, building these trees that I think Danny's about to show you, or just showed you.
Let's take a look, see if we've got I think we've got an iguanodont one in here somewhere.
There is one in there.
Theropods. There we go. Lovely.
Perfect.
Yeah. Fantastic.
So here's a nice like graphical version where we have pictures of all the different types of animals we're talking about here. But the important thing here in terms of the data are these little black lines, right? Showing how these things are related to each other. And so talking about phylogeny, right, and creating these phylogenies, there's a lot of terminology.
Yeah. But, it can seem a little arcane, like there's a lot of jargon here.
Yes.
And we're going to do our best to walk you through that without it seeming too complicated. That's kind of the idea, is just to talk a little bit about, like, what does this tree mean? How do we interpret it? How do you read this kind of thing?
Yeah, how do you read it? And then going a step back, like, how do we actually make these trees and figure this out? So I'm actually going to start up here at the top, right? And whenever you see a split like this, right, we have one line coming up and then it's like diverging. So you have a line going over here to the Saurophinae and a line going over here to the Lambeosaurinae. So we would call those two groups sister taxa. I don't know why it's feminine, but that's what we call them.
Yeah.
Sibling taxa, sorry.
You're good.
So these are, at some point they had a common ancestor back here that has diverged in a couple of different ways.
So they both came from the same species, essentially. That species split at some point, and we get
exactly. So you can think of them kind of like cousins.
Yeah.
Right.
That's a good way to put it. Yeah.
And then you go out and you have more and more distant cousins as you go outward in the tree. So if we go back here, right, Eotrachodon had some common ancestor with both, with that common ancestor of the Saurophinae and Lambeosaurinae, right? And we call that whole group Hadrosauridae, right? So if we're talking about hadrosaurids,
bring this a little closer so they can see in detail.
Yeah. So if we're talking about hadrosaurs or hadrosaurids, right? It's all of these. So think about boxes in boxes in boxes.
Yeah.
Right? Or I feel like
I use Russian nesting dolls sometimes as an analogy.
Or I feel like this is like a phrase that gets bandied about in like junior high sometimes, I feel like. But it's applicable broadly, right? All dolphins are whales, but not all whales are dolphins.
There we go,
right?
yeah.
Dolphins are sort of a box, right, within the larger group that is whales.
Yeah. All toads are frogs, but not all frogs are toads. Yeah.
Right? So we see this over and over. You can apply that same kind of phrase.
Yeah.
To a lot of different groupings. And so, kind of coming all the way back here, we have this largest group, or kind of what we would say these days in phylogenetics, we would say the most inclusive group.
Right, yeah.
Which is the Ornithopoda. And of course, you can still zoom out and go further back from that, but what we're interested in here, we're looking at Ornithopoda. And so some of the early diverging versions of that are things like Thescelosaurus and Hypsilophodon. But keep in mind, when I say early diverging, that doesn't tell us how old it is, where these animals lived, all in the geologic time scale. It tells us how long ago they branched off from everything else.
Sure. So it's kind of relative in a sense. Like if we're looking at hadrosaurs, then these guys are early diverging.
And you can make versions of these diagrams where you show like the actual geologic age of these different animals at the same time that you're showing these relationships. So we would call that like a time calibrated phylogeny.
Right.
But just to note, right, that this doesn't always line up perfectly with geologic time. And so it's not like these animals are ancestral to these up top. It's just that these are the earlier splits within the lineage. And then sometimes you have really rapid evolution along a branch and things get real different. And sometimes like they're in their niche, they're doing their thing, and there's not a lot.
They just kind of look the same for a long time.
Right? Like, even though there might be like actually different species, there is some amount of genetic drift, but the actual what we call phenotype, how the animal looks, doesn't really change substantially.
Crocs are a good example of this. How they look prehistoric.
They fit their niche really well. Gars actually as well.
They actually do have a really slow rate of evolution.
Right. There was a paper out on that recently about how there is actually slower sort of evolutionary change along that lineage.
They don't mutate very much for whatever reason.
I guess they're in their niche. They fit. They're doing what they're doing, and it works. And so there's no real pressure on them to change.
Right. Yeah.
So that pressure being natural selection. So that's kind of our end goal here. So I guess then the next thing is, how do we make these trees? Or are there questions that people want to address before we move
Nothing
on
yet,
to that?
I don't think. People are following along, which is really cool.
All right.
Again, I'm so excited for this. Like, I always jump at the chance to talk about phylogeny, and you're actually having a phylogeneticist with us is really cool. So we really appreciate your time here, Karen.
Yeah.
This is really neat. So when we're talking about this is not some arbitrary arrangement.
No.
So this is actually based on numbers. Like, there's a quantitative aspect to this, which might seem really complicated, but we're going to try and walk you through it a little bit.
Yeah.
Again, this is kind of an advanced topic, so it's really cool that we can discuss this. Yeah.
So this is like grad level class stuff
really
right
is,
here.
yeah.
But we're not going to go into quite that level of detail in the amount of time we have, but we'll talk about it a little. So, in order to figure this out, I get the question a lot, do you use genetics and all? And the answer is, for me, no.
Right.
Because we don't have genetic data.
There's no DNA for any Mesozoic dinosaurs. DNA doesn't last that long, so we just don't have it.
Yeah, exactly. Yeah. But you can create phylogenetic trees based on genetics in a few different ways.
Yeah.
But that's not what I do.
Yeah.
So just, I'm putting it out there. That is the thing that you can do, but not what I do.
Right.
And not what
we don't have that tool
scientists.
available to us as dinosaur paleontologists, so we have to get a little bit more creative, I guess.
Yeah. So all we have to go on are the fossil, mostly, and occasionally some soft tissues.
Yeah.
So what we do is called morphological phylogenetics. So we are using only the morphology, the shape of the animals, the anatomy of them, to determine how they're related. So we're looking for clues in the anatomy that say, here's a feature of the skeleton, right? That appears in, say, these animals, but not any of these.
Sure.
Right? So that would be kind of, uniting, the Sauropophinae and the Lanbiosaurinae.
Be a derived character.
Yes,
Yeah.
exactly.
Yeah.
Exactly. And so what you do is you kind of go through the entire skeleton and you find what we call, which can be broken down into character states. So it could be, if there's a thing that nothing else has, then the state might be that it's, whatever the feature is present in these.
So give us an example.
Absent in these.
Like for hadrosaurs, something that they have that early iguanodonts don't.
So let's see. A good example for them would be actually, it's a little fuzzy where exactly this disappears, but these guys do not have a thumb.
Right,
The iguanodontians, quite famous for their thumbs.
Yeah.
But somewhere in these hadrosaurs, we're not quite sure because we don't have enough fossil hands, they lose both the thumb spike and the carpal bones along with it, actually. So these guys don't really have wrist bones. Sometimes we find just a little disc of probably what was cartilage.
I didn't know they don't have carpals. That's wild. Shoot.
Yeah. So it's like the iguanodontians fuse up the first digit, the thumb, with all the carpal bones, and then it's like they get rid of all of that as a unit. It's like they're all bound together.
That's wild. Yeah.
So when they lose the thumb, they lose the carpals too.
Right. Yeah.
That feels like a Just So story to me, but that's kind of how I think about it.
It could be like a developmental thing.
Yeah.
I don't know. I don't know anything about Evo Devo.
Yeah.
But so interesting. So if you were to find some sort of ornithischian dinosaur like this, and you don't know if it's a duckbill or if it's a more basal iguanodon, that's one of those characteristics where if you find it, there's no carpals, there's no thumb spike. Might be a proper hadrosaur, a duckbill.
Yeah, and that's a really strong character. Just it's a thing that's there or it's not there.
Right.
And those are great. We really like those characters.
Yeah.
Now there's other things, right? Like, and you can maybe kind of tell from this image, but like if you look at Parasaurolophus, it's got these really long, slender forelimbs, and let's say Iguanocolossus down here, it's kind of chonky. Yeah. So, and like having looked at these bones in the forelimbs, I can say, hadrosaurs have a much more long and thin forearm as compared to Iguanodonts. Right, as opposed to robust.
Right,
so you can do characters that are right. So you would say like the length versus the diameter of the radius is, and I honestly don't remember these numbers, but it's more than 8 or it's less than 7, and ideally you want a little bit of a gap there to really, if there's something in between, then
Right. Yeah.
So yeah, so there's different types of characters that you can use. All right, and like I said, the sort of the strongest and the best are just, you know, there's a thing that's there, or it's not there.
Right. So that kind of goes with the question we're getting right here.
All right.
How do you know it's a new feature and not an old feature that got lost in the others? Yeah.
Careful research and looking at a lot of fossils and knowing what they have and what they don't. Yeah.
But that is a challenge for sure. Good question.
For sure.
Yeah.
For sure.
That's something that we encounter.
Yeah. But yeah, I think it's a good point. If you all want to do sort of an
Oh yeah. About this.
I have an example. So with the caveat that pasta is a thing evolved.
Right. Not in like a biological sense, at least.
We're gonna practice making exactly.
Yeah.
We're gonna practice making a little character matrix using pasta because that's something you guys can all immediately understand the different features of.
Right.
You don't have to know anatomy of dinosaurs to do this. So what I have here, and also, and we just raided our little camp kitchen here, and pulled out all the different kinds of pasta we have. And what I'm going to ask chat to do here is to give me ideas about like different features you think that differentiate these different kinds of pasta. So we've got some fettuccine here with some nice wide flat noodles. I'll try and like show them to the camera in case you're not familiar with these types of pasta. Yeah. We got fettuccine. We got good old spaghetti. Okay. This is brown rice spaghetti actually, but you know, spaghetti shaped noodles. All right.
So those are just conical and very, very long.
Cylindrical, right? We got some shells.
Oh, sorry, cylindrical, not conical.
It's all right. It's the afternoon. All right, we got some shells. We got some macaroni. All right. Now we're getting fancy.
Ooh.
We got some cassavece.
And those are an interesting shape.
Right. So they're
like a cylinder that's been slit and then it's kind of curling around.
Yeah, it's kind of got a twist to it and like sort of a hollow, but not like a full tube. So that's different. I don't know how we're gonna handle that. All right.
It's gonna be tricky.
It's gonna be fun. We got some gemelli here, so they're like super twisty, and also with kind of like an actual hole down the middle, I think. Huh. All right. And then weighting the box down currently. I got one more in here.
You can put that box on the ground.
Yeah. Here's for me. We got some gluten-free
nice.
penne in there. So, all right. So hopefully you all know what penne looks like. There's a little window in there. We got some tubes with some ripples.
Nice.
All right. So with that in mind, I'm going to ask chat to
here we go. Interactive portion.
Suggest some characters we could use to kind of like define and differentiate these pastas.
So Kodali says surface texture is one.
Okay.
But with that, we need
Yeah. So like if it's ridged or not.
Yeah. Okay.
So that's a good example of the difference between a character and character states.
Right.
So the way I would write that out, I'm going to call this character 1. All right. I'll try and do this for the camera. All right. So we got character 1. We're going to say. I'm probably going to have to write it.
You're good.
I'll show it.
Kind of put it over your shoulder almost right
perfect.
here.
Yeah. Oh, this marker is just not doing well.
Shoot. We could try a different one if you want. Yeah. Apologies. Those have been sitting in a hot car for
All right. There we go. All right, we're gonna write in green. We've got surface texture. And the way I would write this out then would be, I'll say smooth. And we would code that as a 0 or ridged. And we would code that as a 1.
Interesting. Okay.
All right.
So it's 0s and 1s. It's, you know, for anybody who's ever done, I don't know, anything to do with like really, really basic level coding, you know, you're dealing with 0s and 1s.
And that's basically,
sort of. Not really.
But essentially, But essentially, what we are going to do, what we're trying to do is sort of break down the anatomy into a series of, like in a giant table.
Yeah.
of zeros and ones, and that's
Right.
going to allow us to run it through sort of certain computer algorithms that are then going to try and find the best phylogenetic trees.
Right.
So that's really the game here.
Okay.
All right.
Cool.
Do we have other?
Yeah. So we had hollow or not.
Okay. Lovely.
Yeah.
All right. So I would call that. Let's see. What would we call that character? I'm gonna make it a little biological and call it and just say coelom.
Okay,
Right?
nice.
So in biology, a coelom is basically when, like us, we have a body cavity. So I'm gonna say coelom, and we're gonna make that. I'll kind of show it to the camera. So we're gonna say present or absent. All right. So basically. All right, and we could make it a little more pasta oriented and just say tubular or something, right?
Sure.
But
Yeah.
is it tubular or is it not? Right? Is that present or absent?
Let's see, other kinds of characters. Let's see here. We could do color, possibly.
Yeah, color is a thing you
Or we could do like
do.
maybe material.
Yeah,
Made of wheat? Is it made of
right.
something
We
else?
do in fact have some gluten-free options here, so it's not all
Right.
wheat. So we could say something like composition, and we could go wheat for 0. And then let's see, we got brown rice spaghetti, and then what's this gluten-free? With 4 grains. All right, so I'm gonna go.
Wheat
I'm
or
gonna make
non-wheat?
this. I'm gonna make this a 3-state character. So we're gonna go
shoot.
wheat 0.
Uh-huh.
All right, I'm gonna go. I'll say rice is a 1,
Uh-huh.
and then we'll say multigrain. Is a 2. So it's not all 0s and 1s. You can use multi-state characters.
Uh-huh.
And that there's a couple choices to do with that, right?
Uh-huh.
You could make it ordered, which would basically mean it's going to cost you 1 step to go from 0 to 1 and another to go from 0 to 2. Or it can be unordered, where like no matter what it is,
Sure.
moving back and forth between these different states, it only costs you 1 step evolutionarily.
Okay.
All right, so I'm gonna run out of room on our little board here, so I'm gonna show real quickly
Yeah, how you would
kind of
actually...
how you then would translate this, and maybe I'll only do a couple of our pastas, just for simplicity. So let's
Sure.
do spaghetti,
Okay.
and we'll do our macaroni
Okay.
and our gluten-free penne. So I'll just call that penne. All right. So these are our different species.
Sure.
All right.
Yeah.
And so those are going to go down the side, and then we'll have our different characters across the top. So we just have 1, 2, 3 characters for now.
Okay. And the
Okay.
characters are these different things that we've
Yep.
just outlined, yeah.
1, 2, and 3, right? So here's our little matrix going, and now we can go through and code them. So our spaghetti has a smooth surface texture, so that's going to
Okay.
be a 0. And it has no coelom, right?
Okay.
It is not hollow, so that's going to be absent.
Okay.
And then its composition, our spaghetti is that's the brown rice. All right, so that's going to be a 1.
Okay.
All right, our macaroni has a smooth surface texture.
Okay.
It does have a hole in the middle.
Oh,
so
okay.
That's a 0.
Yeah.
All right, and that is our standard wheat pasta. All right.
0. Okay.
So that's all 0s across.
Triple zeros.
Triple 0s. And then our penne has that ridged surface texture, and it has a coelom, and the penne was that multigrain. All right.
Okay.
So character 3 right now we've got 3 different things going on. And
So now we could basically take this and would you have to pump this into a computer in order to be able to generate a family tree of these pastas?
I mean, let's
Or can
see.
we do that by hand here?
With only 3, I might be able to work it out by hand.
Nice. Okay. Yeah.
So So yeah, so now what we're gonna do, like now I might actually go back and erase our characters just because
Sure.
we're running out of room here.
Yeah.
Oh, I had some all-purpose toilet
Nice.
paper in my pocket here. All right, not all-purpose, guys. It just
Tactical paper.
Yeah. All right, so we're done building the matrix. Now we're gonna use the matrix to build the trees. And you know, this is gonna be kind of simple, but essentially
I can hold it in place if you want.
Yeah. But essentially what we want to do, you know, we want to figure out how these are related to each other, and maybe honestly we don't have enough different kinds
Sure.
here, but
This is just for the purposes of demonstration. It's
just-
going to be really simple.
exactly,
Yeah.
it's a little demo. So if we kind of go through these characters, right? Character 1, you know, spaghetti and macaroni are more similar. Penne is the outlier.
Right, yeah.
For character 2, spaghetti is the outlier.
Huh.
And for character 3,
They're all
different.
Yeah.
So if we sort of take macaroni as our baseline,
Uh-huh.
because it's got zeros, I don't know if that actually makes sense, but you know.
Sure.
But we'll
Would that be our basalmost taxon
yeah,
here?
if we
Okay,
so
okay.
if we root the tree on that,
Uh-huh,
right,
okay.
which rooting the tree is making an assumption, right?
Uh-huh.
That that is the earliest diverging taxa.
Sure.
But if we start with macaroni, then what we would find is if we kind of do the tree like this. All right, so for macaroni, you basically got 2 steps, 2 changes to get to spaghetti. So you've got to change characters 2 and characters 3. And let's see, and you've also got 2 changes to get to penne. You've got to change character 1 and character 3.
Okay.
So it's really kind of equidistant.
Gotcha. Okay. Wait, are we generating a polytomy right here? Is that what you're saying?
Yeah. That is what's happening. I didn't.
It happens sometimes, yeah.
I didn't plan this, guys. So in this case, basically, we have a tree that looks like this, right? We've got
That's not really what you want, but that's okay.
yeah. So we can't really differentiate these. Now,
Uh-huh.
what actually happens in the analysis, though, because that's sort of the end result, right?
Uh-huh.
What actually happens in the analysis is it's going to find a bunch of different trees that maybe have the same length. And here, to be clear, I'm talking about using a particular algorithm called parsimony.
Right.
So parsimony is people. Some people say it's a model that doesn't make any assumptions, but it does. I mean,
Sure.
the assumption here
Is the simplest thing is the thing that
yes.
is
The
way that
assumption
works,
here is
yeah.
the simplest
Yeah.
explanation
Right.
is what is true. That evolution has taken the fewest number of steps or changes between these different things in order to get to where it is.
Right.
All right. Now that's not necessarily true, but it's not.
In most cases,
it's
like
not a bad model, and
Yeah.
and it's no model is perfect, but models can tell you something.
sure, yeah.
So basically, if we actually ran this through a parsimony algorithm, if my brain is doing this right at this late point in the day, basically, we find a few different trees here. So basically, we probably find a tree where we have like. So I'm just gonna do this.
There we
right?
go, yeah.
Yeah. So where we have macaroni coming off, and then because it's basically like kind of equal steps to get from there to either spaghetti or penne, right? You would just sort of have like you could have one tree like this where you have spaghetti and then penne. You could have another tree. We really should have done 4 because that would make it work better, but
Yeah.
and then you could have another tree where, let's see. I guess maybe I just shouldn't root this. I don't know.
We were planning to have like a great big whiteboard space for this because we were going to try to do this on the side of the trailer in camp, and
Yeah.
then couldn't do that. So
That wasn't going to work. So
Right.
we're on a tiny whiteboard. So
Yeah.
apologies. I had a grand plan here, but
this is good practice for next time.
We're improvising. We're making
Yeah.
it work. So if you flip spaghetti and penne here, it wouldn't actually mean anything on the tree. So that's something else important about trees is like when you have like a
node
branch
there.
point like this,
Yeah.
you can flip where these two are relative to each other, but it doesn't make any difference,
Sure,
actually.
yeah.
It's still spaghetti and penne are more closely related to each other than macaroni.
Right.
Right.
So there's still sister taxa right there, and then
Right.
macaroni is the out
So
group.
that's what that's saying. But actually, let me see. Yeah. I made like.
It's going to be an imperfect analogy.
I made
so.
I made a tiny matrix here that basically is uninformative, right? Like no.
And sometimes this happens when we've
Right,
got
no,
very
it
few
is. Actually,
specimens
it's a-
with very few characters. Like,
Right.
this is not uncommon in paleontology.
I mean, we would have a lot more than like 3 and 3, a
Yeah,
3 by 3
yeah.
grade. I think that the current matrix I'm working on is something like 60 taxa and like 600 some characters.
This is the big ornithischian
Yeah.
phylogeny that you're working on right now?
Yep.
I mentioned that at the beginning of stream when I was over there, and that's
Yeah.
really exciting.
Yeah. So like I said, this is a very simplistic version of what we do, and yeah, I certainly could not calculate that thing by hand. That is stuff that computers are good at.
Sometimes very, very powerful computers are necessary for that kind of thing.
Yeah. Parsimony analyses are usually pretty quick.
Yeah, but if you get into like Bayesian
If you're
stuff,
doing Bayesian or maximum likelihood, which are basically different models that are making different statistical kind of choices about and like modeling like the actual evolutionary rate, so you could say like early on the evolutionary rate should be slow, but actually then it should pick up as things really go into their niches, so you can really fine tune stuff with Bayesian analyses. There's a lot, phylogeneticists will argue about this stuff so much in so many ways. My general thoughts on this are that it's good to use a couple different models. So I typically run my analyses both in parsimony and in a Bayesian analysis.
and if they both come to the same conclusion,
Right.
then
Well, and like, so if you have things where,
Yeah.
if we went back to like that larger tree, right? If you have certain groupings that show up in both the parsimony and the Bayesian analysis, then
That's a real
you're
thing.
it's not an artifact of the model you're choosing.
Right.
It's actually what the data say. Now, that still goes back to, have you picked good characters?
Right, yeah. So again, that's tricky. Like when we're trying to come up with that for pasta, you can see how difficult it is for something really simple like this, but think about if you're dealing with different bones within a really complex organism like a dinosaur.
Right. And you're
Yeah.
dealing with a whole bunch of different bones. Like I said, pasta is a really fun exercise because it's fairly straightforward. And like I said, it doesn't make any assumptions that any of you know anything about anatomy. Some of you may, but some of you may not. We can all talk about pasta.
Yeah.
But it kind of gets you thinking about breaking down the specifics of that shape. How do I describe that shape and turn it from
Something that's qualitative into something that's quantitative.
Exactly.
Yeah, you're putting a number on it essentially.
Yeah. Yeah, so I guess I don't know. I think that's kind of the basics, but I'm happy to.
Let's see, we've got a few, I think. Have we heard of Mirasaur? Yeah, that just came out yesterday. La Petite Prince. We'll talk about that later, maybe. People are making jokes about Pastafarianism.
Absolutely. Yeah. That is appropriate.
But this is one of those concepts where it's important to understand if you're going to study evolution. I wanted to just kind of give an introduction to everybody so that you know that when we're making those phylogenies, those family trees like that, it's less arbitrary than you would think it is. Like, that's the thing, is that these are really complex systems.
There's a lot of thought and a lot of work that goes. It's not
Absolutely.
a drawing. Now, actually, I say that, but the one in the book is sort of a compilation of several different people's work, and ultimately just sort of a drawn, best representation of best agreement between everybody's work that's out there.
When we're looking at a scientific paper, though.
Yes, when we're actually looking at a scientific paper, that's not just it's not a hypothesis. Well, it is a hypothesis, but it's not like something we just thought up and drew the tree. Right.
There's a lot of steps that go into that, and those should be outlined in the paper itself, often in the sub-info or something like that, the supplementary information.
Sometimes it's supplementary. I mean, I think it should be in the paper.
I agree.
But but it should be, and some people will describe a new species and they'll just say, we're using the data matrix from this other paper and we just added this taxon. Here's the character here's the string of numbers for just this species, just tack it on.
Sure.
And that's fine. But then if you're actually doing like a big phylogeny and a new one with new characters and all that stuff, then you'll have this big like supplementary data file. And it used to be that they would be in a big table at the end of the paper.
Right.
But that's not really practical. And if you're going to actually try and use that data, so now it's typically just supplementary info.
You can download that and you can make your own.
And it's like a downloadable file that, if you have, usually the program that most scientists use for this is Mesquite.
So Mesquite is a standard.
Yeah. I mean, people may use other things, but that uses what's called Nexus files. It's a standard file format. That's what people use.
Gotcha.
You know? Yeah.
Can we bring back the phylogeny again in the book?
Yeah.
So the difficult thing about you turn almost right to it.
Yeah.
Perfect. When we're trying to code different dinosaur taxa into a tree like that, is that often you're dealing with incomplete information, and it's incomplete in like varying ways. Where like you're not going to have a complete skeleton for all of these guys.
No.
So some of them, like Iguanodont artensis, you've got dozens of skeletons that are complete from Belgium. But then you look at something like Ioni right there. There's I think one skeleton that's known. It's pretty good.
It's pretty complete.
Yeah.
But like so Iguanodont over here, that's really complete, known from a lot of skeletons. Iguanocolossus.
Oh, sorry, I was pointing at the wrong guy.
It's okay. Iguanocolossus is not super complete. It's important. It's from Cedar Mountain, all that stuff, but it's it's got a pelvis, a few skull bones. And, it's not very complete. And here we go. Great example. Planocoxa.
Yeah.
Planocoxa is like an ilium and limb. And so, and if you're trying to compare that to, say, an animal,
you're gonna have trouble.
It's really hard. So it's really important to have these sort of keystone taxa, like Iguanodont, where you actually can code the whole skeleton, and then other things will sort of slot in relative to that.
Right.
And there still may be places where it's unclear how certain animals are related to each other, and that's how you can end up with these what we call polytomies, which I guess I can actually draw out.
Yeah, a polytomy.
Yeah.
So polytomies are what you're trying to avoid when you're making a tree. Like if you keep getting polytomies left and right, it means that like either you've got incomplete information or maybe you're just kind of doing something wrong.
Yeah. So basically, right here, if this is true, right, what it shows is like macaroni, right, branched off first, and then spaghetti and penne are more related, and that definitely seems wrong, right? But we didn't do enough characters, all right? There's something wrong with our analysis. But if you got a polytomy, it would basically look like this.
They're all equally related to each other.
They're all, so basically what that says is we're actually not sure which thing branched off before the others.
Right.
And there's nothing inherently wrong with a few polytomies.
Right.
Like it just means that there's uncertainty. But yeah, if you just have a giant polytomy with like 20 taxa all coming off the same,
a problem. Yeah.
No, then it's kind of like, oh well, we don't know anything, do we?
Right. Yeah.
So that's not great. You have 3 or 4 taxa coming out in a few places. Like that's not a big deal. Not in paleontology.
Yeah. Now, I want to ask too, I'm always thinking about ontogeny. What role does that play in making phylogenies, and how can it mess things up if you fail to account for it?
Well.
So ontogeny is the study of how organisms change during their lifetimes. So like a frog going from an egg to a tadpole to a froglet, frogling? Froglet? To an adult frog. Like, it's going through a lot of changes. It's got a tail at one point,
And then later it doesn't.
Right.
So how do you code that?
Yeah.
And so one thing that you can do, and look, people that do phylogeny for amphibians, it's kind of a whole other thing. And they'll usually specify characters like for the ontogenic state, right?
Sure,
Like tail present or absent, like as a tadpole, tail present or absent, something like that, right? So they'll basically specify which stage of the animal's life cycle they are talking about. And code those separately, but like all in the same line for a given species. With dinosaurs, it's not always perfectly clear, like
Yeah.
which taxa are juveniles and which are not. Though sometimes we definitely know that we have juveniles. See my paper on Ayuku.
Right. Yeah.
Right, and so this was actually, you know, one of those projects that my PhD advisor Kathy Forster had been working on describing this new species from South Africa that's only known from babies. And she was like, all right, you're going to do the phylogeny, and you're going to figure out how to do the phylogeny on only these babies. And so the problem is, I'll give a good example that I think humans can relate to even, is number of teeth.
Right.
Like when we're little, we don't have all the molars that we have as adults. You know, our baby teeth have fewer teeth than our adult teeth. And dinosaurs are constantly shedding and regrowing teeth, but a similar phenomenon happens where younger individuals have kind of a shorter jaw and fewer teeth in it as compared to the adults. But that's also something that changes through phylogeny. Like these guys up here have something like
500
30,
plus teeth?
yeah, something like 30 to 40 tooth sockets just in like each, like one side of the lower jaw, right? And the upper and the other, right? So they've got like just a ton of teeth. Whereas like iguanodont has, I don't know, something like 25, you know, and these guys down here have like 12. So it is something. We don't want to ignore it because it does have like an evolutionary signal, genetic signal.
Right.
And so the simplest way to deal with that is just to figure out which characters in your evolutionary like analysis also change through ontogeny. So anytime you have a taxa with an ontogenetic series, that's really important because that can tell you like which of those evolutionary characters are also changing through ontogeny. And then you just have to be really careful with those characters.
Right.
And basically what I did was I was just very explicit that I was trying to code adult, or at least close to adult character states.
Right.
And so for this baby taxon that we had, anything that changed through ontogeny in closely related species, I would just say, you know what? That's a question mark. Oh yeah, because you can also have question marks with all those zeros and ones. And in fact, in paleontology, much of our matrices are question marks. So you get. So I would just say, you know what? I don't know what the adult state is. I assume that it is not 4 teeth in the maxilla. That's what we have in this juvenile.
Right.
So I'm just going to write that as a question mark.
However, if you say that you weren't being careful, you have this dinosaur Ayuku, it's a baby, but you don't realize that. You think, oh, it just looks really weird. I'm going to plug it into the tree and assume that it's an adult. What's likely to happen when you do that?
It's not going to go where it should.
Usually it'll be pretty basal on the tree,
Usually
right?
it will end up like branching sort of earlier. It will end up in a more inclusive clade than you expect.
Yeah. So it'll look primitive, to use like old-fashioned words here.
Or to use more phylogeneticist words, plesiomorphic.
Yes.
But weirdly, that's not always what happens. I had some early runs with Ayuku analyses where it would actually like jump up the tree in places. Like it was weird. It had, I don't know. I got that stuff sorted out. But my point is, you know, it's not always gonna. The babies are not always going to end up looking more primitive or plesiomorphic or branching earlier. In fact, sometimes they actually just end up jumping up the tree for weird reasons. So, I don't know. And I honestly don't have a good explanation for why that was happening.
That is really unusual.
It was I kind of have a handle on it, but it's esoteric and arcane. So,
yeah.
Because
that has happened with other dinosaurs, like I don't know, to use an example that always kind of annoys me, Raptor Rex, which is a tyrannosaur described from anyway. It's probably a juvenile Tarbosaurus, Raptor Rex, probably.
Yeah.
But it came out as super basal on the tree. It's like, oh, it's this weird late surviving primitive thing.
Right. And no, it's just a juvenile.
Exactly. Yeah. So
and that's a thing that happens a lot. So that's definitely the more common thing that happens.
There are buffalo now.
Yeah. This is really cool, Karen. It's really cool talking to you about this. And we look at a lot of phylogenies, a lot of family trees. throughout our streams, so it's cool to be able to talk to somebody who actually does this, professionally. You are, there's very few people who, not very few, but it's a specific thing within paleontology to be really good at phylogenetics and understand all of the ins and outs.
There's a lot of serious statistics that go with it,
Yeah.
and kind of verging into computer programming, which honestly I'm not good at that part. But
Yeah.
but at least working with sort of like command line programs and really setting your parameters specifically and telling the program exactly what you want it to do, and understanding the statistics that come from that. So people out there who are really into like that kind of thing, this is a fun aspect of paleontology.
This is a way that you could get into paleo potentially.
Yeah,
and I know we've
absolutely.
got some coders in the chat.
Yeah.
So
Yeah.
what are some, if somebody were to try and learn phylogenetics, if they're inspired by this broadcast right now, what should they look into? What's a program that they should start with, or a tutorial, or what would you recommend?
If they want to start messing around with stuff, I would recommend TNT.
Yeah.
That's a parsimony-based analysis. It runs really fast. You can get a free version for download.
TNT better than POW?
Yeah.
Yeah? Okay.
I would recommend TNT. And if you want to read more about it, like textbook style, Inferring Phylogenies by Felsenstein is sort of the go-to textbook for that. It's
Inferring
textbook.
Phylogenies.
Inferring Phylogenies
By
Felsenstein.
Felsenstein. Okay.
It's dry and it's statistical. I'm not saying it's a fun light read, but
Sure.
if you're really interested in this and you want to learn more, that's definitely the place to go.
Cool.
Yeah.
Cool. Yeah. I at some point really want to put together like a grand phylogenetics stream or series of streams where I talk about this and show examples, and we like run through some phylogenies and on stream in TNT, I guess. I was thinking about doing PALP, but it's been a long time since I picked up any of these programs, and I'm glad to have that recommendation.
Yeah,
TNT
is very straightforward, and like I said, an advantage is you can download it for free.
Nice.
Thanks. There is a free version thanks to the Willy Hennig Society.
Willy Hennig was an entomologist, I think, who kind of helped pioneer the
he
idea
was
of
definitely the
phylogenetics?
pioneer of like phylogenetics, specifically cladistics.
Cladistics. Yes.
Which is basically parsimony analyses as we do them today. So, and a lot of the terminology that we use was set forth by Willy Hennig.
Right.
So
Yeah, cool. Thanks so much, Karen. This has been a lot of fun. Really appreciate it. I'll put this away, and then we've only got a few minutes left till we're going to wrap up here in the quarry, so I'll walk around and answer some more questions and stuff. And but big thanks to Karen for walking us through this. Really appreciate it.
Yep.
All right. Set this down over here. And there we go.
No, this is open. It's all good. No. You just put it back.
Yeah, pasta in my brain.
Pasta tonight.
I wouldn't mind pasta, shoot.
You say lol.
I'll get water in a minute when we're done. But yeah, pretty cool to be able to talk about this with somebody who, sort of a leading figure in that field, dinosaur phylogenetics. Again, Karen is working on this grand phylogeny of ornithischian dinosaurs. That's going to be really big when it comes out. And there are some potential surprises in there, things that folks might find shocking. She's not done with it, but if the findings that are coming out right now still hold in a few months when she's done with this, it could shake up the dinosaur family tree a bit. And I'm excited to see that. Shoot. Yeah. And never would have thought of the pasta analogy. Isn't it a good one, Golganek? Shoot. I'm, with Karen's permission, going to see if I can steal that for when we do a big phylogenetics series of streams. You'll look up maybe when Willy Hennig's birthday is. When is that? Let me check that out. Willy Hennig, German biologist and zoologist, founder of phylogenetic systematics. He's looking very stern right there. And anyway, he was born April 20th, 420. Oh boy. Died November 5th. I don't know if an anniversary stream would be good, but whatever. We'll find an excuse to talk about cladistics and talk about this stuff in depth in the future. But let's see if we've got any other questions from the chat right now, just about general stuff, before we wrap things up here. And 7 days before Tapir Day, says Lenina. There you go, April 27th, World Tapir Day. And no, Death Source, Velociraptor is not going to become an ornithischian. But, yeah, I don't know. I've already said too much. We'll talk about that another time. That'll be a cool paper to discuss once it drops, because the dinosaur world will be abuzz. People will be all in a tizzy over that paper when Karen publishes it, if she consistently keeps finding those same results. Yeah. And the more the better for the clade, says Jody Fish. Well, that's the thing. We could do that, like, we could fire up TNT and actually do that with a bunch of different pasta shapes and varieties when I'm back in the office at some point. I think that would be pretty neat. And I'm also interested in doing this just so that I better understand it. I'm going to have to do a lot of background reading on this kind of thing. Maybe ask Karen a few questions about how these particular things work. But in order for me to be able to explain this stuff well, I've got to be able to do it myself, and I've got to understand it fully. And so, yeah, I think this is going to be a really cool exercise for me too. Yeah. And Ms. Art Rules says, any living dinosaurs? Ms. Art Rules? You know that birds are living dinosaurs, Ms. Art Rules, yeah. Yeah. And a collab live stream. It's possible, Jody Fish. We'll see. Karen's pretty busy during the fall. Busy teaching schedule when she's teaching anatomy labs and stuff. Again, Dr. Karen Poole teaches in Arkansas at the New York Institute of Technology School in Jonesboro, I think. But yeah, so she's teaching medical students, physician's assistants, people like that. The doctors of the future, she's teaching them anatomy. So it's important stuff. And she was telling me how many other paleontologists she works alongside with there. There's a good cadre of paleo people at that school, studying fossils, but making their living teaching anatomy to medical students, which is really neat. I talk a lot about how there are many, many different ways to be a paleontologist, and that's one of the more common ways, is to teach med students anatomy, and then also pursue your own research in fossil science. So, yeah. But yeah. And who's the pro, says Dr. Poole, is the goat, I heard. I think she is. And a cadre, beautiful word. Thank you. Yeah, I like that one. And yeah, Neil Shubin is a good example of that, Golganek. A fish paleontologist who, for a while, he was head of the anatomy lab, I think at the University of Chicago. Yeah, we've watched that clip from Your Inner Fish many times. Great documentary, and it helps illustrate that point. Some of the most known paleontologists today to med school students, which is, I think it's pretty cool. I feel a certain degree of pride in that, like, yeah, you know who knows anatomy best? Paleontologists. Especially when we're talking about osteology, the anatomy of bones. But when that's all you've got, when you don't have genes, you don't have DNA from extinct organisms, you got to do gross morphology, the morphology of the bones, osteology, sometimes soft tissue, stuff like that. You got to get those skills really well honed. You've got to know that subject really, really well, because that's the tool that you have when you don't have DNA that you can study. So, yeah. And, yeah. And dinosaurs, humans, frogs, it's all the same bits. This is true, Charlie's Dragon. This is true. That's a subject we talk about a lot too, this idea of homology. So, you know, look at our arm and hand. I think I just talked about this the other day, but we'll talk about it again. We've got phalanges, we've got metacarpals and carpal bones in the upper part of our hand and our wrist. Then you've got radius and ulna in your forearm. And then humerus is your upper arm bone. That's the same exact structure that we see in things like frogs, that we see in things like dinosaurs, that we see in things like mammals. A whale and her big flipper, she's got the same exact bones that we have. They might be very differently shaped, but there's still one bone, the humerus, two bones, radius and ulna, a bunch of little bones, the carpals and metacarpals, and then you got the phalanges. And that's not a coincidence. And it's not anything mystical or magical. It's that humans and whales and dinosaurs and frogs, we all share a common ancestor. Just like that phylogeny that we were looking at with the iguanodontians. You know, you've got ancestors and descendant. Well, it doesn't show ancestor-descendant relationships, but you know, basal and derived. It's the same thing, where if you trace, you could make a big phylogeny where you put humans, frogs, dinosaurs, whales. We could run a phylogeny like that, a cladogram, where we look at different characteristics and we find out who is most closely related to whom. And if you do it correctly, you know, you can approach it from a bunch of different angles, but you should get humans and whales next to each other, because we're both mammals. We share a most recent common ancestor. And whales split, and then after that, you would get dinosaurs. Dinosaurs are much more distantly related, but they would be the next one out. So dinosaurs link up with mammals about 320 million years ago, something like that, back in the Carboniferous period. And then after that, you would have frogs, which diverged even earlier than that. So yeah, again, the whole point of a cladogram like that is to show who is most closely related to whom. And you look for shared characteristics in order to be able to determine that. So this is a subject we'll be talking about more in the future, and I'm excited for it. It's going to be cool. It's one of these things that very few people in the general public understand, not because it's overtly difficult, not because it's that difficult, that hard to understand, but just because it doesn't really get taught in schools. Because in order to do that, you have to talk about evolution. And a lot of people are really scared of that concept, especially in the United States. So it doesn't typically get taught until you get to college. Even though it's the most fundamental, important concept in all of biology, it gets pretty much ignored unless you happen to take a biology course in college. There are people here in the chat who have, learned all about evolution in middle school and high school, and that's awesome. But then there are people, very smart people, mind you, very well-read, savvy people who are maybe never been exposed to these concepts just because it's not widely taught. Unless you go to college and happen to take a biology course, you're just not going to be introduced to it. So that's one of the reasons why I do these streams is if you don't understand evolution, you don't understand biology. And if you don't understand biology, you don't understand the history of life on Earth and where we belong as human beings in the grand scheme of things. These are important subjects. And understanding them better, I think, can proffer a certain degree of perspective and, dare I say, wisdom. If we as human beings want to continue existing on this planet and not completely wrecking it, if we want to make sure that we're not causing a mass extinction of all these other critters that we share this planet with, you have to understand how they're related to one another. You have to understand their place in the ecosystem. You have to understand how all these things are connected to one another. It's important. These are pertinent things, So anyway. With that, we're going to wrap up here. Let's find somebody to raid into here. Who else is live right now? I'm going to go help with closing up the quarry for the day. Let's see. Scrolling through. Looks like Shark Geek is on. Although they're doing some just chatting. Let's see if there's anybody doing science. Whoop, hang on. Oh, Draw Curiosity is streaming right now. Duh. Let's go see what they're up to. Oh, and here comes some crazy wind. Shoot.
Take everything down.
Yeah. Okay, well, let me wrap this up, and I can help out. Twitch app is being super buggy. Stop it. Oh, don't do that. Quit it. There we go. Channel, chat, raid, draw curiosity. Excellent. Well, everybody, thank you for being here today. Again, short stream. That wasn't my choice. Starlink was down for a solid hour and a half. Made our stream, oh, actually almost. So, I don't know. When it works, it works. When it doesn't work, that causes problems. And it wasn't working today. Hopefully, everything is resolved and we can have a full-length stream tomorrow. We'll see, but it's field work. You never know. Anyway, thank you everybody who tuned in today. Thank you for your questions, for your curiosity, for your attention. Thanks for talking phylogenetics with us. It was a lot of fun. And again, big thanks to Karen Poole for taking time out of her day to teach us a thing or two about figuring out evolutionary family trees. Thank you to the moderators for keeping this a safe and welcoming community for everybody. Thank you to those financial supporters, subscribers, gifters, cheerers, all of them. Couldn't do this without you. Thank you to all the question askers and even those lurkers. Appreciate everybody who tuned in today. And with any luck, we'll be on regular time tomorrow. So take care, everybody. Let's go right into Draw Curiosity. I'll see you there.